Hi everybody, I'm Bijou Allard. Today I'm going to be talking about my perspective as a student who has used open science resources in my coursework. And this is my presentation, Seeing Ourselves as Scientists, a Student Testimonial on Open Access Neuroscience Research. Before I get started, I want to acknowledge the wonderful people that collaborated with me on this project. So my fellow undergraduate students at the time, Mariela Rodriguez and Nicole Marie Duran were amazing collaborators on this project, as well as Dr. Caroline Wilson, who as an educator represents, I think, some of the pedagogical principles that we were just talking about. She's been absolutely incredible. So I'll get started. So I'll start by talking a little about myself. I just graduated from Chapman this May with my bachelor's degree in psychology, thank you, and a minor in neuroscience. I'm currently living in Portland, Oregon, so just a little bit south. Don't recommend taking the train 4 hours each way in one single day. It's going to be a long one for me. But I'm living in Portland. I currently work for a nonprofit called Northwest Habilitation Services, where I support individuals with severe and persistent mental illness in their activities of daily living. Now I'm going to tell you a little bit about Chapman's neuroscience minor, which is where the project I'm going to be talking about was completed under. So the neuroscience minor at Chapman was established in 2020. It's a 22-credit program. It's interdisciplinary. Students are required to complete coursework from psychology, philosophy, biology, computer science, and statistics classes. Additionally, there's two kind of core classes that are specifically neuroscience focused, Neuro 301, which has a lecture and lab component, as well as a capstone class. And so pictured there's a little picture of some of us minors at graduation all together, so that was lovely. All right, as an undergraduate student, I've done my fair share of in-class and outside-of-class research sources, and I've noticed a couple challenges that come especially with in-class research opportunities. The first is that they can be really, really resource intensive. So in class, especially in my minor program, we did not have access to wet lab resources for the minor. All we had was an anatomy lab that we were working out of. And so we had anatomy tables, but that was about it as far as equipment that we had access to, maybe a couple EMGs, but we didn't have access to really complex EEGs, things like that. And so these kind of expensive facilities can pose a really big barrier, I think, especially for institutions with limited funding. I can only imagine like community colleges and things like that. You're just not really going to have necessarily the resources to support large-scale wet lab, especially kind of research. The next is that I think there's a lot of diverse research interests in one classroom. This is definitely true in neuroscience. Neuroscience is extremely interdisciplinary, and you have people coming from all kind of majors and minors in my minor with extremely diverse research interests. At my university, most of our neuroscience research faculty are primarily computational, but not a lot of us were necessarily interested in doing computational research. And so that can be a challenge in engaging students interest-wise if the research you're doing in class is not necessarily aligning with their particular interests. I think the final and one of the most kind of important barriers is that there's such limited time. The project I'll be talking about today was completed over the course of 5 weeks as part of the class. Even if you have a full semester to do a research project, it's really, really limited in what you're going to be able to get done, let alone you're not going to be able to deal with an IRB or that kind of approval process during that time. So that really adds an extra layer of challenge to the whole thing. And a great way to kind of navigate these challenges is through the use of open science resources. So today I'll be talking about 2 resources that I use in my neuroscience course. One is the Allen Institute's resources, and the other is the Chan-Zuckerberg Cell Gene Database. All right, so let me just give you a quick overview of the project timeline that I worked on. So to begin, we started by exploring open science resources in class. So these included completing 2 adaptations of Allen science resources. The first was the cell types, transcriptomics, morphology, and electrophysiology activity, and the second was the brain donation and bioethics activity. After completing those 2 entry sources, we formed groups. Not all groups use open science resources, as I'll talk about later, but we formed groups based on our interests of what tools we wanted to use in our research. We also conducted a literature review as a group and then formed a testable hypothesis as well as a research question. Thirdly, we collected and analyzed data. So for our group using open science tools, that consisted of a lot of data extraction from the open science databases, as well as statistical analyses and how we're going to interpret those results in the context of previous research. And then finally, we had a synthesis and presentation of findings that included a 250-word abstract that our groups wrote together, as well as a creation of a conference-style research poster and poster presentation in a mini in-class research symposium. All right, to begin, I'm going to talk about a little bit of the foundational materials we were given to kind of orient ourselves with these open science resources. The first was the cell types, transcriptomics, morphology, and electrophysiology activity. We completed an adapted version of this. We did not do the full activity necessarily, but this was a really valuable introduction to some of the tools used in neuroscience. A lot of times we might not know the kind of diverse array of tools that are used to get this data, like electrophysiology, or what different types of microscopy look like, and these kind of things. So this was a really good entry resource for us. And secondly, and I think honestly most importantly for me, was the brain donation and bioethics module. In this module, we talked a lot about what it means to donate your brain to science, how scientists get data about cells, especially human data, and what are the people behind that, and what does that look like, and how do we behave ethically with that data as researchers? And for me, that was really, really valuable, and I really appreciated that being a core focus of our course. And yeah, I'll talk about that a little bit more later on. So for my group, we knew we wanted to use open science tools. That was something that we all came together agreeing on. After doing those introductory resources, we thought, wow, this is a really diverse pool of data that we have to work with, and we really want to do something with it. We also knew that we really wanted to do something related to dementia and neurodegenerative disease. That was a big kind of topic of conversation in our class. We had a few guest speakers come in, and that was something we were all passionate about. And our professor lovingly presented us with some research that she had recently found, linking microglia with dementia, especially Alzheimer's disease. And this is kind of a newer route of research that's gaining a lot of traction recently, and especially in neuroscience. And we thought that was really, really interesting. And while we were exploring the Zuckerberg-Chan database, we actually found that they had some data on microglial cell expression, especially gene expression within microglia. So that led us to come up with the research question: how does microglial gene expression differ between people diagnosed with Alzheimer's disease and those without dementia? And is this same pattern observed across other diseases related to cognitive impairment, such as Parkinson's disease and Lewy body dementia? Okay, so for our data collection, it was actually relatively simple for us. This was, as I mentioned, an in-class research project. So we went to the Zuckerberg-Chan database, and we filtered under the gene expression data set. So we wanted to look for genes expressed by microglial cells. So then we filtered from the gene expression to just microglial cells, and from there further filtered down to data related to dementias, and you can filter by all different kinds of neurodegenerative diseases. So this was a figure that automatically populates when you narrow down in this Zuckerberg-Chan database, and this figure is really, really helpful because these little dots right here indicate how much gene expression is happening based on each of the disorders and what percentage of microglial cells are expressing those specific genes. So just from this figure here, we kind of had a really nice entry point to what we were going to be able to extract more from when we pulled the data. And we could see that there was some pattern definitely emerging, and that some genes were being much more highly expressed than others within certain neurodegenerative diseases. So after this, we exported our data as CSV files. That's one of the awesome things about these open science databases, that they're super accessible in CSV, and there's a lot of resources about how to export and work with the data after it's been exported. And so that was really helpful for us. And then we used JASP, which is also an open source software, to analyze data and obtain kind of general descriptive statistics. All right, and this is kind of the final figure for my main research poster. This is displaying the proportion of gene expression across these different genes organized by what disorder is here. And from this figure is very complicated and hard to interpret and very busy. So from there, we looked at the patterns that we observed, and we decided to narrow down on a few specific genes after doing some further research into what had already been looked at within these specific genes. So we targeted approximately 6 different genes that had been linked to Alzheimer's disease as well as other dementias in prior research. And we wanted to really focus in on those. So from there, we created another figure, which was a table here, that is kind of a more descriptive thing looking at the proportion of microglial cells across those disorders that are expressing those specific genes. And even just from this table here, you can clearly see that there's something different about Alzheimer's disease. And we noticed that right away, and we saw that Alzheimer's disease seems to be exhibiting a distinct expression profile that's specifically related to microglial genes, which are these ones, that are linked to the inflammatory response. Now, this is a pattern that has been heavily echoed in previous research linking microglia and Alzheimer's disease, And so this was a really cool discovery because us as students got to kind of make this discovery that had been going on in research for ourselves and see it play out in the data set. And so that was really cool. We saw this pattern and we went back to research and we uncovered some of the mechanisms as to why that might be happening. Honorable mention for Parkinson's disease and Lewy body dementia, which had a little bit more association with this gene here, which is related to synaptic neuronal function and cytoskeleton structure. Those are also things we found out in our research. And so this was our final kind of product for a research presentation. This was our poster. It's a little bit different than a standard research poster in that we have our little abstract included here. We were encouraged heavily to create our own figures. And so we used some open resources here to create this little diagram. And it was a really awesome way to just, get experience creating a research poster. I had been lucky enough to have had experience creating research posters prior to this project. However, nobody else in my group had that experience, and so it was a really nice entry point for us as students to get experience making conference-style research posters and creating a conference-style research poster presentation in kind of that elevator pitch style. Okay, and just to summarize, some of the skills that we gained from this activity were 1, forming research questions, conducting literature reviews, interpreting and exploring complex data sets on our own in a student-directed way, and also just learning about open source tools as well as how they can be used to generate unique research questions, and also interpreting results in the context of previous research. I do want to acknowledge some of the challenges with using these databases. For one, it's really hard to narrow down the specific research topic. Our whole course wasn't designed around open source data, so this was just one element of the course, and I think that might have made it a little bit easier if we had had a lot more previous experience working with the data sets. We might have more of ideas about what they had to offer and what was out there. Also, especially for the Zuckerberg-Chan database, accessing information about the donors was a little bit harder, and that's something we really appreciated about Alan, is that it was a little bit easier, but that was something that I would love to just learn more about, and it was something that we had to really dig into the data to find out. And also, I want to acknowledge that this is something that you definitely need prior computer science and statistics knowledge if you're going to do. We all had at least had computer science introductory course before this course, and at least a statistics introductory course before this course, and many of us had higher level data analysis courses as well. So that's something that I would definitely want to keep in mind if I was an educator. Like, people need to have some kind of foundation in order to actually be able to successfully analyze data. And yeah, I want to share some of our reflections. I interviewed some of the other people who I worked with on this project, and it was really cool to talk about how this project impacted us. Nicole said that it was really cool to see how many different approaches you can take to looking at one set of data and how much analysis can just come out of one data set. Mariella also said something that I really appreciate, is that a lot of times science is paywalled, and we realize that, especially as undergraduate students. Especially I'm realizing that now, losing access to all my open data that I just now, I realize a lot of it is paywalled, and that's one really nice thing about these open science sources, is that they are accessible and they can be inspiring, because I'm like, oh, I still have access to this den of data, even if I'm not connected to a university right now. And for me, it was really cool to see more about collaboration. Obviously, we collaborated as a group here, but we also, the collaboration that's required of scientists across the world to make these databases possible. And I think that that's a really important point. So some of the takeaways I have is, one, these open science activities made science really accessible for us. These were free resources. There's a lot of activities available, especially through Allen, to familiarize students with these data sets, as well as they provide an ability for, provide a way to conduct research on a reasonable time scale, which I really appreciate as a student. They also kind of empower students to build independence and design their own projects, which can make the project a lot more interesting, and also give people more confidence that, oh, I can do research on my own, and maybe I'll go out and do more research after this. And finally, they're just really creative. They encourage students to create and present data in novel ways and try to do something different with a data set and data that's already been collected. And I just want to do a quick honorable mention for some of the impacts that this has. I mean, a lot of this is due to the neuroscience minor. It's a really well-designed program at Chapman, but we've all gone on to do research in various capacities. So Mara is currently working at Chapman School of Pharmacy. My other colleague, Nikki, she is now running an NHS-funded biomedical study at Chalk Children's Hospital in Irvine. And the summer after this course, I actually completed an NSF-funded fellowship at the University of Michigan Graduate Neuroscience program, where I studied molecular neuroscience and the superior colliculus, which was a really awesome experience. And yeah, this is a slide created by my professor, just highlighting that a lot of these open access materials promote the core competencies that she uses for her research design and classroom design. And they also don't require necessarily students to have a deep expertise of a specific field of study. It's just a kind of a great way to get people's foot in the door and a way to explore topics that they might be passionate about in a way that's really classroom accessible. And if you want to ever contact her, I have included her information on here, and I'm happy to share these slides with you later on. And yeah, I just want to say thank you guys so much, and now I'll be answering questions.
All right, our mic runners will be coming around. I expect many enthusiastic questions. This is exactly the kind of experiences from the educator perspective that we want to be creating.
Okay, good.
All right, we got a question in the back.
Okay, awesome.
Hi, Bijou. That was amazing.
Oh, thank you.
Well done. Congratulations on some great work there. I wonder if you could speak to how you guys work together as a group to execute the project, to come up with the poster, and the roles of each participant within the group. And if you have any tips on how your instructor facilitated your group coordination, that would be great.
Oh my
Thank
gosh.
you.
Yeah, absolutely. So this is something I didn't even have time to touch on in the presentation, but Dr. Wilson uses team-based learning approaches in all of her courses. I took another course with her called Applied Human Neurophysiology that was entirely team-based, but this project had a lot of those team-based learning principles. If you're not familiar with that, I would highly recommend that. It really changed my perspective as a student with group work because we had groups that formed earlier on in the semester, and then we kind of worked with those groups over time and built more of a connection with them. It was really helpful too because our class was cohort-based, so a lot of us, we all had to complete this introductory neuroscience course together, and then we completed the capstone together as well as many other courses. And so that allowed us to all get to know everyone in the class, which I think made the process of doing collaborative science work a lot easier for us. So if that's at all possible within your course, I think building those connections early is a really good way to do that. But yeah, I think we didn't necessarily have assigned roles, but it was just nice that we all had connections with each other prior to this project.
Thank you so much. That was a fantastic talk
thank you.
and awesome work. I actually have I really appreciated that normal was in quotes.
Yeah.
You just acknowledged so many things, and I just really appreciated that. I was curious, so you talked, I really appreciated your reflections and how you talked about the benefits of using open source resources. When formulating the research question, though, as you've already acknowledged, there's a lot of challenges. So my question has two parts. One, how did y'all as a group decide on pursuing open source resources? And two, how were you introduced to open source resources? So specifically, did your instructor do anything to aid that introduction, or was it self-started?
Yeah, absolutely. So as I kind of mentioned, we completed as a class some of those introductory activities provided by Alan. But in addition to that, we used a lot of actual figures that my professor had pulled from the Alan database in order to illustrate our points. In our class, we didn't have access to a lot of microscopy work. We had only a few slide samples, and not all of it was very applicable to what we were studying. And so we used a lot of the database to actually look at how certain genes or where certain neurons were expressed in the brain. And so that was kind of an entry point. As far as forming the research question, that was definitely challenging. I think once we had an idea of something that we were really interested in with the microglia and neurodegenerative disease, at that point we thought, okay, we're gonna have to find a way to look at this. And we had a lot of kind of storming around it. Luckily, my professor, she guided that process of forming the question and thinking, okay, what's the scope that's gonna actually be possible within our 5-week timeline that we have? But I think we just had that initial spark of inspiration, and from there it was just a whittling down process and coming with a few iterations and checking in with the professor as well. But yeah.
We have time for one more.
So that was wonderful and so articulate and really interesting for me to hear. I, through this program called, this NSF-funded program called Step Up, as a PhD student, was able to create and teach a course. And seeing this was kind of interesting because I tried my very hardest to basically do this exact course at UW. And I have a poster that will talk about that. So anyways, you as a student just are so articulate and thoughtful on the whole process. So I love that, and I would hope that maybe some of my students could be like you up there on the stage. I was really curious, like a couple questions, how much you knew about open science before the course, and what about like just the course itself, maybe the course title, course description that caught your eye? Like, was it you wanting to learn about open science? Because like sometimes that's a little bit less of like the exciting thing, or was it more like knowing that open science allows you to work with data and get to do a project? I have a million sub questions, but I will cut myself off there. But thank you again.
Yeah, so I think for me, I was just really interested in the neuroscience minor in general, and this was one of the core courses involved in the neuroscience minor. And I think something clever that Dr. Wilson did was she kind of just snuck in the open science into the course. She didn't necessarily advertise it as like, okay, this is going to be a course where you use open science. It was just implemented within it. And I think part of what also drew me to it was that Dr. Wilson herself was very open about her pedagogical techniques and why she structured a course in a certain way and why she used these certain strategies and why she was introducing us to these open science resources. And so I think for me, that made me think of them in a different way and think of them as like, oh, this is an opportunity for me to explore something that I actually don't get a chance to access at my university. And I, for my whole undergrad, was considering doing a PhD in neuroscience, and I wanted to get that molecular experience, but I didn't have access to a molecular lab until I was at U of M. And so once I saw this resource, Dr. Wilson was like, oh, this is a way for you to experiment with that data, and that was what really sparked me to get into it. I also just care a lot about accessibility in science, and so I think that was something our course talked about and something that was important to me and that I saw within the open science data.
Cool, thank you.
Yeah, of course.
Thank you
Thank
for
you.
an amazing presentation.
Oh, thank you.